My name is Dr. Caitlin Mullarky. I'm an associate professor in biochemistry and biomedical sciences at McMaster University, and I'm a viral immunologist by training. So today we're going to answer your questions about viruses. The first question we have here is, what exactly is a virus and how is it different from bacteria? Viruses are microorganisms, and they are a lot different than bacteria in that viruses are very small. So most viruses we can't see with a naked eye or even through a light microscope. We need really powerful microscopes to visualize them. And then a really important way that they're different than bacteria is that viruses actually need to get into a cell in order to make lots and lots of copies of themselves. Taking a step back, viruses are what I like to call obligate intracellular parasites, which sounds like a mouthful. They are infectious agents that can transmit from one host to another, whether that host is a human, an animal, a plant, a bacteria. They need to get into a cell of that host in order to be able to make more copies of themselves and then to spread to a new host. So this idea that they need a cell to make more copies of themselves, much different than bacteria. Bacteria are capable of independent replication and they have independent metabolism. Are all viruses harmful, or can some actually be helpful to humans? Now this is a really good question because viruses are all around us all of the time. The vast majority of them have no impact on human health, either good or bad. They're completely neutral. We tend to focus on the ones that make us sick, those ones that we hear about the most, but that's really a very small fraction of all of the viruses that exist. So, no, not all viruses are harmful. Some are, and they range in how harmful they are to, let's say, human health in particular. Obviously, they can infect lots and lots. Well, viruses can infect any living thing. Can some actually be helpful? Yes, there's evidence to suggest that viruses are actually important for the development of both our immune system and maybe some anatomical features in human development. There's plenty of symbioses we can point to in the environment. This is moving beyond humans, where viruses are required in order for either two organisms to survive or to complement an ecosystem. So that there are definitely ways viruses can be helpful to humans. I'll also point out that we can engineer viruses for therapeutic interventions, so for medical interventions. We use viruses for vaccines. We are developing viruses for cancer therapies. Even if viruses inherently aren't good for humans, again, most of them are neutral, there are ways that we can manipulate them to actually be beneficial to humans. Can you give us an overview of the viral life cycle? The viral life cycle starts when a virus infects a cell. And for animal viruses, the most common way this happens is the proteins on the surface of the virus recognize certain receptors on host cells. So the virus will bind to those receptors, and then it can either be taken entirely into the cell, or it can inject its nucleic acid, its genetic material, into that cell. Now, once the virus is in the cell, it's going to make lots and lots of copies of itself. That is one of the main differences of how viruses are distinct from bacteria, again, because they need to get into the cell, and also because the number of virus particles that will be produced from that infectious cell is exponential. One infected cell can produce tens of thousands or even millions of virus particles. So when viruses replicate, the analogy I like to use is they replicate in an assembly line fashion. They make all of their different parts, and they make lots and lots of copies of all of those different parts, and then they assemble those parts into the virus particle. If you think about the way we put cars together, you start with a frame, then the doors go on the frame, and then the wheels, and so on and so forth. It's the same idea with viruses. They're going to make copies of their genetic material. They're going to make proteins that offer that protective shell, and they'll make lots and lots of copies of them and then assemble them together. Once the virus particle is assembled, then it's ready to leave that infected cell and transmit to a new host. The important point of a virus in the context of an infected cell is that when it's making all of those different parts, whether it's proteins or it's nucleic acid, it's using host cell machinery. So viruses actually leverage all of these processes and machinery that that exists for the host cell, and they skew it to make copies of themselves. And that really helps us understand why fighting viruses can be challenging, and also developing drugs for viruses can be challenging. The next question actually has to do with my own work in influenza virus vaccines, and what some of the biggest breakthroughs in that field are. Influenza virus vaccines are a really interesting vaccine because they have to be reformulated every year. And this is pretty unique to vaccines. We do see the COVID boosters being updated more regularly, but the vast majority of vaccines we are not updating on an annual basis. And the reason that we have to do that is because the virus changes. It evolves quickly such that the vaccines from last year no longer protect against the strains that are circulating come the next flu season. It's also the case that it takes us a long time to produce those vaccines, so it takes at least 6 months between the time we pick the vaccine strains to when those vaccines are rolled out. So some of the biggest breakthroughs in the field of influenza virology, I think, are going to be sort of in two camps. The first is in technologies, right? If we can shorten that 6-month time frame such that we can produce vaccines on a shorter, more constrained time frame, then we don't have to worry about those times where new strains, we have to worry less about the times where new strains come up. We'll be able to ramp up vaccine production. We'll be able to be more flexible in shifting between strains, and that can make a big difference in terms of getting vaccines out to the population. The second is the types of protection these vaccines offer. So typically our seasonal flu vaccines offer limited protection. They only protect against a few strains. The idea in the influenza virus field is it would be better if we had universal influenza virus vaccines that protected against ideally all strains of influenza. So the universal influenza vaccines would provide greater protection against strains. Maybe we could have flu vaccines now that we only had every few years, or every 5 years or every 10 years. There's a lot of active research around being able to produce vaccines that provide broader protection against more influenza virus strains. The last question is, at McMaster, you play a big role in student education and mentoring. What advice do you have for future scientists interested in virology? My biggest advice is just to be curious. Our knowledge of viruses is constantly evolving. We learn new things about viruses all the time. The fact that they are omnipresent and they infect all living things means that we're constantly surrounded by them, and we are going to continue to learn new things about viruses and the interface of viruses and our immune system.
Thank you for watching. A special thanks to Dr. Caitlin Mullarky for her insightful answers. If you enjoyed this video, please consider leaving a like and subscribing to the channel for more content.